Hi, folks. Welcome to the 2024 National 5 Biology paper. This is section 2 that we're going to be covering in this session. So let's rattle right on to question 1 and have a look at this. So section 2, as always, is worth 75 marks, and you have to attempt all questions. Hint number 1 from a biology teacher: do attempt all the questions. Don't leave things blank, even if you're not sure, because you never know if your guess is going to get you a mark or not. So question 1, Euglena is a single-celled organism found in water. The diagram shows some of the structures within the Euglena. We've got the chloroplast, we've got the cell membrane, and we've got another structure A. You have to identify structure A, which is the nucleus. Okay, next one, give the function of a chloroplast. So again, this should be a straightforward one for you if you know your cell organelles and their functions. That is the site of photosynthesis for the chloroplast. Have a wee look at 1.1 in your cell structures and definitions if you're not sure about those two. It suggests why Euglena is not a typical plant cell. So there's a couple of reasons. First of all, it does not have a cell wall, which we find in a typical plant cell, and it has no vacuole. Okay, so the Euglena has no cell wall and no vacuole. They're both features which we tend to find in plant cells. Next question, the average body length of a Euglena is 50 micrometers. They can travel 1.3 body lengths in 1 second. Calculate how many micrometers a Euglena could travel in 30 seconds. Okay, so for the calculation here, we know that the body length of the Euglena is 50 micrometers. We know they travel 1.3 body lengths in 1 second. So we need to work out what 1.3 of 50 is. Okay, so we times 50 by 1.3 and we get 65. Okay, so that means that it travels 65 micrometers in 1 second. However, the question asks us how far can it travel in 30 seconds. So we're going to do the 65. We're going to then times that by 30. And we will get 1950. Okay, 1950 micrometers for that one. Okay, so hopefully you followed how we worked that one out. That's a wee bit of a problem-solving question there, isn't it? On to question 2. A group of students measured the mass of pieces of turnip tissue before placing them in different concentrations of salt solution. After 1 hour, the change in the mass of the turnip tissue was recorded. You have to predict the percentage change in mass in 2 grams per 100 centimeters cubed of salt solution. Now, for this one, what we have to do is we need to look at the table and we can see that for a concentration of 1 and a concentration of 3, we know the percentage change in mass. So for a concentration of 2, we're going to go halfway between, because 2 is halfway between 1 and 3. For the percentage change, we're going to go halfway between 1 and 17, which is going to be 14. Because the change in mass can either be positive or negative, we would have to put a positive, a plus before the 14 there. So any value between 11 and 17 would be accepted by the SQA, but I'm going for 14 because it's right in the middle there. Part B, identify the salt solution where most of the turnip cells would be turgid. So if the turnip cells were turgid, that would mean that they'd gained a lot of water. And we know that plant cells become turgid when they gain a lot of water. So if you gain water, the mass of the cell is going to increase. And so we're looking for the concentration that causes the biggest increase in the mass of this turnip cell. And that, if we look at the table, is going to be plus 17, which is the concentration of 1 gram per 100 centimeters cubed. Okay, the pieces of turnip used in this investigation all had a starting mass of 6 grams. Calculate the final mass of the piece of turnip in the 8 gram per 100 centimeter cubed concentration of salt solution. So if we have a look at this one, we can see that the percentage change in mass is minus 9%. Okay, so what they're saying is at the start, it's 6 grams. When it goes into this concentration, it loses 9%. What will be the final mass? So we basically need to find out what 9% of 6 is, and then take that 9% away from the original 6 grams. So to work that out, there's a couple of ways to do it. You can go 6 divided by 100. That gives you 1%. Okay, so 6 divided by 100 gives you 0.06. That's 1%. We then multiply that by 9 to give us 9%, 54. And then we get our original value, which was 6, starting mass of 6 grams, and we take away 9%, which we've worked out as 0.54, and we should get 6 minus 0.54, we should get 5.46 for that one. So the correct answer, the final answer, is 5.46. Now, the SQA would allow you to round that up to 5.5. Okay, question 3. We're really motoring now. The diagram represents the processes that generate ATP in muscle cells. So hopefully we recognise aerobic respiration and fermentation there, which go in slightly different pathways depending on whether oxygen is present or absent. So let's have a look at the questions. Part 1, we have to name substance J and product K. So substance J is the product of the breakdown of glucose, okay, and the breakdown of glucose produces 2 molecules of pyruvate. Product K is the waste product produced at the end of aerobic respiration. Now we look at the other two, we've got carbon dioxide and ATP listed already. So the one that's missing there is going to be water, okay? So pyruvate and water. If we know our respiration reactions, then we should know those, okay? If you don't know them yet, I would suggest having a good look over 1.6 and memorizing those reactions so that you do know them. Okay, part 2, state the number of ATP molecules that would be produced from each molecule of glucose during fermentation. So we know that for fermentation, there is no second stage producing ATP. In aerobic respiration, the second stage produces a large number, but in fermentation, there is no ATP made. The only ATP made during fermentation happens during the first reaction, where glucose is broken down into pyruvate, and as well as 2 molecules of pyruvate, we also produce 2 molecules of ATP. Okay, again, that's just a memory one. You need to know that 2 ATP are made in that first breakdown reaction. Part B, name the cell structure where aerobic respiration is completed. So this is a throwback to 1.1 as well, to cell structure, and hopefully we remember that the mitochondria is the site of aerobic respiration. Okay, so that's what we're looking for there. Part C, it's a suggest question. These are the tougher ones. Suggest why a muscle cell might carry out fermentation rather than aerobic respiration. So what are the conditions for aerobic respiration? Well, we require oxygen to be present. So if a muscle cell carries out fermentation instead, that would suggest that there is no oxygen present, or oxygen is absent would be acceptable as well for that one. Okay, so well done if you did well on that question. If not, have a wee look over topic 1.6 again. Question 4. Potatoes store sugar in the form of starch. Glucose 1-phosphate, or G1P for short, molecules are converted to starch in an enzyme-controlled reaction. This enzyme can be found in potato extract. Important, that sentence. Keep that in mind for later. So if we look at the diagram, we can see glucose 1-phosphate is being broken down by the enzyme into starch. In fact, I shouldn't have said broken down. It's actually been built up, isn't it? Because we've got the small glucose 1-phosphate molecules have been built up into one larger molecule of starch. And that's important because the first question asks us, identify the type of reaction carried out by this enzyme. This type of reaction is called a synthesis reaction. That's where we build up smaller molecules into one or several larger molecules. Identify the substrate in this reaction. Well, the substrate is what is acted upon by the enzyme, and the product is what is made at the end of the reaction. Okay, so in this case, it will be the glucose 1-phosphate, which is our substrate, and the starch, which is our product. Okay, so G1P, or glucose 1-phosphate, you can write out the full thing if you like, is the answer there. Part B, an experiment was set up to investigate the time taken for starch to be produced. All previously made starch was removed from the potato extract. 2 rows of a dimple tile were set up as shown in the diagram. At 5-minute intervals, the content of the dimples at each column was tested for the presence of starch. The results are shown in the table. So we can see there's 2 rows. The first row has G1P, that's our substrate, and potato extract, which, if we read back to the top, is where we get our enzyme. So substrate plus enzyme equals an enzyme-controlled reaction. So therefore, we would expect to see a change in that first row there. In the second row, we have G1P in distilled water. Now, this time, we've removed the potato extract, which means we've removed the enzyme. And without an enzyme catalyzing that reaction, nothing is going to happen. Okay, and we can see that based on the results. The G1P in potato extract eventually detect a change and detect that starch is present, so the reaction's taking place because starch is our product. And the G1P plus distilled water shows no change, no starch is present, so therefore no reaction has taken place. Now that we've done all that analysis before we've even got to the questions, because It's important to understand what's happening in the experiment because it will make answering the questions a bit easier. Okay, 4b part 1, suggest a reason for any previously made starch being removed from the potato extract. So we want to focus on starch being made during the experiment, okay? So that means that we've got to remove the starch so that only starch made during the experiment would be detected. We do not want starch that was made before the experiment contaminating our results or mixing up our results, okay? We have to prove that the starch was actually made during the experiment and not before. Part 2, state 2 variables that should be controlled to make this experiment valid. So let's have a look at the experiment. When I am teaching variables to my class, I always get them to remember the acronym MTVC, which stands for MTV channel. Now, when I was a youngster in the 80s, MTV was the best channel to watch. It was a music channel, and we all watched that. That when the number of channels we had were much more limited than now. So MTVC stands for mass, time and temperature, volume, and concentration. Now, if you can remember MTVC, you're going to remember the bulk of these variables we have to name in this question in the exam. Now, when we're dealing with solutions here, we're dealing with liquids, right? We're putting them onto a dimple tray, so it's liquids. And therefore, if we're thinking liquids, we're not probably thinking mass. They mention the time already in the experiment, so we're not thinking time here either. They don't actually mention the temperature here, and they don't mention the volume, and they don't mention the concentration, okay? Now, with the G1P, that is one solution. So we could say the volume of the G1P or the concentration of the G1P. We've also got potato extract, which is the enzyme. So again, we could say the volume of the potato extract or enzyme or the concentration of the potato extract or enzyme. And lastly, we've also got distilled water in this experiment. So for distilled water, we could say the volume. We wouldn't necessarily say the concentration because that should be the same, because if it's pure water, that concentration is just not going to vary. So the volume of, or the concentration of for this question, okay? And you could say any of the things, so the G1P, the extract, or the water for volume, and similarly for the concentration, we're saying G1P or the extract, or the SQA do allow you to say the enzyme instead of the extract for that one. Okay, so that is what they're looking for there. Those are the variables that would be controlled. Now, why is we mentioned temperature a moment ago. Why? Temperature is not mentioned in the experiment, but why is it not an acceptable answer here? Because simply controlling the temperature in the lab, in the room, is very difficult to do. You can't really control the temperature. You can measure it, but it's very difficult to control it. So therefore, that's why it's not allowed here. The volume and concentration we can control. Okay, part 3, 4b part 3, explain how the results show that the enzyme is required for the reaction to occur. Well, simply put, if we look at the results, when the enzyme is present, a reaction occurs, starch is produced. When the enzyme is not present, i.e. we've got distilled water instead of the extract, there is no reaction, okay? So, starch is produced when either the extract, or you could say the enzyme, is present, okay? That would be fine. Or you could say something like no starch is produced in the second row, okay? And no starch is produced in the control experiment, because that's a control experiment, remember, where we've replaced the factor we're investigating. That's the enzyme, and we've replaced it with an alternative that will have no reaction, which is the water. Okay, moving on to question 5. The concentration of lactate in the blood of a long-distance cyclist was measured while they were cycling at different speeds. The graph shows these measurements at the start and the end of the competition season. Okay, perfecto. We've got the lactate concentration up the side here, and we've got the speed that they were cycling and the start and the end of the season. And we can see the start of the season has got a slightly greater upward curve, especially at those higher speeds and compared to the end of the season. So anyway, we'll see what the questions are asking us. Part A, what was the lactate concentration at the start of the season when the cyclist was travelling at 40 kilometres per hour? So start of the season is the solid line, and we go to 40 kilometres an hour, and we move up to the solid line, which is here, and we read all the way across the graph, and we can see that's sitting at around about 2.6. And we know it's 2.6 because that line's 2, that line there must be 3, because it's halfway between 2 and 4, and because there are 5 boxes between 2 and 3, that makes each box worth 0.2, and it's the third box up, so it's 2.2, 2.4, 2.6. Okay, so the answer is going to be 2.6 units there. Part B, calculate the percentage decrease in lactate concentration at the end of the season compared to the start of the season when the cyclist was travelling at 52 kilometres per hour. So it's the old percentage change question. We get one of those in every single exam. So I would say if you don't know this formula, remember it. Okay, it's going to help you so much when it comes to the exam. It's basically a free mark once you know how to do these well. So we're talking about the decrease between the end compared to the start when the cyclist was travelling at 52. So we go to 52 kilometres per hour. We go up to the end of the season, he was at 4. And at the start of the season, we were up at 6.4. So the change would be the 6.4 take away the 4. 6.4 take away 4, which is equal to 2.4. That's the change. We're going to divide that by the original. What was it at the start of the season here? It was at, what was it at? 6.4, wasn't it? Divided by the 6.4, then we're going to times that by 100, okay? So when we pop those numbers into a calculator, because even Mr. White cannot do this in his head, 2.4 divided by 6.4 times by 100 gives us 37.5, okay? So 37.5% is our answer there, okay? So the answer is really irrelevant in terms of us practicing here. The important thing is, do you know how to do the calculation regardless of the numbers or the situation you're given? Well, this formula here is going to help you get on the way and be able to do that. And as I say, there's always a question on percentage change, so it can be an increase or a decrease in every exam paper, so it's well worth knowing that. Okay, 5 part C. Calculate how many times greater the maximum lactate concentration was at the start of the season compared to the end of the season. So let's look at our maximums for the start and the end. We can see they're going to be here. So the maximum for the start was 9. Okay, 9. Go along here, 9. And for the end of the season, it was 5. Okay, if we go along here, that takes us to 5. So it goes from 9 to 5. Now, when you're asked how many times greater something is, we have to calculate how many times the smaller number goes into the bigger number. So it's a simple big divided by small, 9 divided by 5 in this occasion. Okay, 9 divided by 5 gives us 1.8. Okay, big divided by small when you're asked how many times greater something is than something else. All right. And remember, yes, we do our calculation, but we also put our answer on the box that they want us to put the answer in. Okay, number 6. We're motoring along now. The diagram represents a reflex arc when a person touches a cactus plant. Ouchie. Okay, we've got the fingers touching the cactus, we've got the receptor there, and then we've got a big long neuron that goes from the receptor towards the central nervous system. We have to identify that neuron Y first of all. Now, I always teach my classes, when it comes to neurons, remember SIM, S-I-M, which if you're unsure about how to remember it, just remember you've got a SIM card in your phone. I know that none of you will ever forget that you've got a phone. So you get a SIM card in there and that will help you remember the neurons, okay? S-I-M, SIM. So the first one is the sensory neuron. Okay, that's the one that goes from the receptor to the CNS. And Z, we can see from this diagram, is the neuron that goes from the CNS back out towards our effector here that's going to carry out our response. So that's going to be the motor neuron, okay? Because in the CNS here in the middle, we have the interneuron. So S-I-M, sensory, intermotor, okay? And that's an easy 2 marks for us if we remember some. Part 2, state how messages are transferred at the synapse. Well, messages travel along neurons as electrical impulses, but when they reach the synapse, because there's a gap, we cannot travel by electrical impulse. We need a continuous contact for that, so therefore they have to travel across the synapse via chemicals, okay, which will bind to the receptors on the next neuron and pass along the signal from there. So chemicals transfer messages at the synapse. Be careful with this one not to say chemical messengers, because that is the name we give to our hormones, okay? And we don't want to get them confused. Okay, part B, identify the effector in this reflex arc. Well, the effector is what carries out the response, okay? And the response is at this end. So what is the effector? What's the thing that's actually carrying out the response? Now, you might be tempted to say the hand, because it's the hand that's moving, but what's causing the hand to move? That's the key thing. And our hands, or any part of our body, is moved due to muscle contraction, okay? And so we look at the diagram, it's the muscle there that's causing the response. It's causing it's the effector that's causing the arm to move, okay? So You could also, sometimes the effector is also a gland, but in this particular diagram, it's the muscle that's labelled, so that's the one we're going with. Part C, state the function of a reflex. Now, this is a simple memorization question. You should have learned this. If you haven't yet, go away and memorize this, practice it, write it out 6 times until you can do it without even thinking about it, okay? What is the function of a reflex? It's to protect the body from harm, okay? Now, in this particular question, you could say protect the hand, or the finger even, which is the bit that touches the cactus, from harm. So you could be specific about this particular question if you like, but the generic response that you can give to any of these questions is protect the body from harm. So I would suggest learning that one. Okay, that's a bit of memory work for you to go away and do if you got that one wrong. Okay, number 7, the diagram relates to reproduction in humans. We've got various stages happening here, all good. A part 1, we've got name organ G. Now, organ G produces the gamete called the egg. Which organ does that in humans? It is the ovary. Part B, what happens during fertilization? This is a one that students often get wrong, but I always go over this very thoroughly with my classes and try to make sure that they memorize the definition of fertilization so that they don't get this wrong. Now, the easiest way to say this is the nuclei meaning more than one nucleus. The nuclei of the gametes, that's the egg and the sperm, and you could just say egg and sperm if you prefer. They fuse together, and they form a diploid zygote, okay? Now, the SQA will probably let you away with just saying the sperm nucleus and the egg nucleus join together to form a zygote, okay? In fact, in this one, they don't even need you to say zygote. However, if you memorize that whole sentence there, you can't really go wrong when you're asked about this in the exam, okay? And then, funnily enough, the next question is name cell H. Cell H is formed from the fusion of the egg and the sperm, and we know that's going to be the diploid zygote. Now, the SQ again will accept zygote on its own. Why do I keep writing diploid zygote? Because it reminds me that it's a diploid cell. And it's important, I think, that we use diploid and haploid before the cell types as often as possible, because when you get a question about diploid and haploid, it will remind you which is which. Okay, it's a bit of extra detail, but it will help you in the long run. Part B, name the unspecialized cells that can be obtained from an embryo at the very early stage. So the cells that we can obtain from an embryo are the embryonic stem cells, okay? It's the stem cells that get you the mark here. Embryonic is just for a bit of flourish in your answer, but yeah, stem cells is all we need to put. Okay, question 8. Progressive retinal atrophy is a rare condition in some dogs, such as cockapoos, and it can result in blindness. PRA is caused by the inheritance of a recessive form. Recessive, remember, meaning the one that needs 2 copies to be expressed and is usually represented by a lowercase letter, in this case, lowercase r. Depending on their genotype, a dog's phenotype can be described as affected, unaffected, or a carrier. A cockapoo breeder tested the DNA of a female dog and 3 male dogs before choosing which pair to breed. The results are shown in the table. Okay, so we get a heterozygous female there that's a carrier, a heterozygous male who's a carrier, a homozygous recessive male who we'll talk about in a second once we do the answer, and male 3 is a homozygous dominant cockapoo. Okay, so male 2, who's got 2 lowercase r's, it tells us here that the PRA condition is inherited via the recessive form, so the lowercase r. This person only has those lowercase r's, so therefore they are going to be affected. Okay, they are going to be affected by this condition. And the other male is the 2 capital R's. Well, they don't have any genes for PRA, so they are going to be unaffected. Okay, they are going to be unaffected by this. If we have a look at question 2 there, state the term used to describe the genotype of a carrier of PRA. Well, I mentioned it as I was reading through the table myself, and this is why it's good to use these words as often as you can, because when you get into the habit of immediately thinking capital small, heterozygous, then it makes it easy when you come to do the questions in the exam. So, yep, the carriers are both capital and small, 2 different alleles, so they would be described as heterozygous. Remember, homozygous means the 2 alleles are the same. Heterozygous, the 2 alleles are different. Part 3, the breeder selected a male based on their DNA results for this condition. The offspring were as follows. We've got 4 unaffected and 4 carriers, okay? Now, if we look up at our table we did earlier, unaffected means capital R's all round, and the carrier means a capital R and a small r. So which male was chosen to breed with the female to give us offspring that only have those genotypes? Well, the female is a carrier, okay, which is fine. But if we were to use male 1, who's also a carrier, because both of them have one lowercase r, that would mean that some of the offspring would inherit one lowercase r from mum, one lowercase r from dad, and therefore would have two lowercase r's and be affected. So male 1 can't be the father here because some of the offspring would be affected if they were. Similar situation with male 2, because they only have lowercase r's, some of the offspring would be affected. Male 3, however, only has capital R's, which means every one of male 3's offspring will have at least one capital R, which means some of them will be unaffected completely. But because mum, the female, has one lowercase r, some of them will also be carriers. So it's going to be male 3 for that reason. Okay. Question 8b. Another breeder did not carry out DNA tests before breeding a pair of cockapoos. All their offspring were affected by PRA. Give the genotypes of the parents involved in this cross. Okay, so to have PRA, we've established that you need two lowercase r's. Now, if all the offspring are going to be affected by PRA, that means all the offspring have two lowercase r's. Now, if every single offspring only has two lowercase r's, that means they must have inherited a lowercase r from both parents. And if all the offspring are inheriting that, that means parents must only have lowercase r's themselves. If even one of those parents had an uppercase r, then at least some of the offspring would have an uppercase R and would be carriers, okay? So that's why the parents both have to be lowercase R's only. 8 part C, give the term used to describe the different forms of a gene. We've used it a few times already as we've talked around this question. This is just a definition, so a bit of memory work. But the different forms of a gene are known as an allele, okay? An allele is a different form of a gene. So if you didn't know that one, go away and memorise that one. Okay, question 9. Coronavirus is a pathogen which causes COVID-19. Probably most of you will remember that. It was a huge deal a few years ago. Most people with COVID-19 feel better within a few days or weeks of their first symptoms. Long COVID can be diagnosed when symptoms last longer. The symptoms of long COVID include extreme breathlessness, memory, and concentration issues. In some cases, other organs can also be affected, causing other health issues. A study monitored the changes in the health of 500 patients with long COVID. Between the start and the end of the study, the percentage of patients with extreme breathlessness decreased from 38% to 30%. Those with memory and concentration issues decreased from 48% to 38%. And those with other health issues decreased from 57% to 45%. At the end of the study, 10% had no organs affected, 65% of patients had only one organ affected, and 25% had multiple organs affected. So lots of information there from that passage. So what we have to do is sort of digest all that info, and then we have to answer the questions below. This is our scientific literacy question, which we get one of every year now in the paper. Part A, name the type of white blood cell that produces antibodies that are against coronavirus. So we know that there are two types of white blood cells that we've learned about in topic 2.6, and those two types of blood cells are pathogens. They are the engulfing white blood cells that destroy the pathogens, and we've got the lymphocytes, which are the ones that produce our antibodies, okay, those specific antibodies that are specific to one type of pathogen. Perfect. Okay, the next question asks us to fill in a column heading and the relevant data into the table. We have to complete it using the information above. So the heading for this column in the table should include extreme breathlessness, memory and concentration issues, and other health issues. So let's look in the passage to where we can find those things. So I've found extreme breathlessness there, memory and concentration issues, and other health issues. So what are these things? These things are the symptoms of long COVID, okay? So that's exactly what we're going to put into our table. The next one, we have to put in the percentage of patients at the start and end of the study that had each of those things. And if we look at our passage above, percentage of patients with extreme breathlessness went from 38 to 30, okay? To 8 to 30. The memory and concentration issues went from 48 to 38. So we're extracting the correct data there. And the other health issues went from 57 to 45. So that's how we fill out the table. Use information that's already up there. We're not thinking up any new info. We're not having to work anything out. We're just simply extracting the correct information from the passage. Okay, question 9C. Calculate the simple whole number ratio of the percentage of long COVID patients who had multiple organs affected to those who had one organ affected to those who had no organs affected. So ratios can be tricky. In this case, the first thing to sort of work out for ourselves is the order in which we're going to write the numbers in here. So they've got multiple organs affected as the first thing to go in your ratio, followed by one organ, followed by no organs. So we need to put our numbers into that calculation in the same order, even though when in the passage it has those numbers in a different order. It has no organs first, then one organ second, then multiple organs last. We have to switch that order around. So it's 25, 65, and 10. Okay. So let's go down here. We've got 25, 65, and 10. Okay. Now we need to find a common denominator, a common factor that goes into all three of these numbers. Okay, we get 25, 65, and 10. Now, you kind of do this in your head. So quickly I can see that 5 probably goes into all of these numbers, or you can do it in a calculator and work your way down, work out which number you can put into all of them. Start off with, I don't know, whichever number you like and work your way through it. But if I'm going to divide them all by 5, I'll get 5, I'll get 13, and I'll get 2. Okay. So the answers, and we can't simplify that any further, and they are all indeed whole numbers. This is an important part of the ratio question. The most common mistake I see from people is they don't simplify the ratio down enough, or they start using decimals, okay, rather than whole numbers. So that's how we work out that one. And question D here suggests a reason why the study could be considered invalid. Now, I have a little saying in my class where I say VAVA. So validity is related to variables. When they say suggest a reason why the study could be considered invalid, as soon as I see the word valid, I think variables. Okay. Now, if we look at a study like this, study of monitoring the health change in 500 patients, and look at all of the symptoms of this: extreme breathlessness, memory and concentration, other health issues. Okay. How many different things are factors or variables for all of these conditions? Well, COVID isn't the only thing that causes breathlessness. Things like asthma and other restrictive pulmonary conditions can cause extreme breathlessness. Memory and concentration issues, well, you get those as you get older, just naturally, even without a condition. And other health issues can be caused by a wide variety of things. So essentially the issue with this particular study is that there are way too many variables. Okay, there's too many variables to control, and something along those lines would get you the mark for that one. Okay, in A, the diagram represents part of the circulatory system in humans. Okay, so we've got the body, the lungs, the heart there, and all of the vessels going in and out. Part 1, we have to name the chamber labelled M. Now, this chamber here, labelled M. Now, when I'm teaching this to my students, I always say to them, with this chamber, draw the outline of it with a pencil like that. And when you draw the outline of that chamber, it's shaped in the letter V. Okay, so that tells us that this is the ventricle. Okay. However, we also have to state the side that ventricle is part of. Now, in the heart, remember, the left is the right and the right is the left. So as I look at this diagram, that's on the left-hand side. So therefore, that is the right ventricle. Okay. Next one, compare the carbon dioxide concentrations in the blood traveling through blood vessels L and M. Well, if we look at L here, L is leaving the lungs, okay? It's going from the lungs back to the heart. So any blood vessel that is leaving the lungs is going to be high in oxygen and low in CO2. Okay, so L is low in CO2. Okay, what about N? Well, N is coming back from the body, that body which is full of cells that are respiring, using up all the oxygen and giving out carbon dioxide. So N is going to be high in CO2. Okay, so what is the issue here? And what is the comparison? Well, we would say that N is higher than L. Okay, we've compared the concentrations. There is more CO2 in N than L, or vice versa. There is less in L than there is in N. Something along those lines would get you the mark there. Okay, B, the heart is composed of muscle tissue that requires its own blood supply. Name the blood vessel that supplies the heart tissue with blood. Now, this is, again, a straightforward knowledge memorization question. You need to know it's the coronary artery. Okay, that supplies the heart with its own supply of blood, which it needs for the oxygen that it needs for aerobic respiration, so it can produce ATP and the heart muscles can contract. Which leads us on nicely to part B. Explain why the heart muscle cells will stop contracting if this blood vessel becomes blocked. Well, if the blood vessel becomes blocked, the heart cells are going to get no oxygen or no glucose for that matter. Okay, and therefore will no longer be able to produce ATP via aerobic respiration. Okay, we know that ATP is produced via aerobic respiration. And why is that relevant? Because no longer produce ATP via to contract the heart muscles. Okay, so that's why they will stop contracting. They are no longer getting that ATP from aerobic respiration. And why is that? Because aerobic respiration requires both oxygen and glucose in order to produce that ATP. Okay, nice one. We're on to question 11. We're really moving along nicely now. The gas exchange surfaces in fish are called gills. Gills absorb oxygen from water. Suggest two features of gills that increase the efficiency of absorption. Now, this is where my students often go, Mr. White, we've never learned about fish and we've never learned about gills. How can I answer this question? Well, the solution is apply the same knowledge that you used in other similar situations like the alveoli in the human lungs or the villi in the small intestine. As soon as you see the words increase the efficiency of absorption, you should be thinking three things. You should be thinking that it has a large surface area. You should be thinking that it has thin walls. And you should also be thinking that it has an extensive blood supply. Okay? As soon as you see those words, efficiency of absorption, think those three adaptations, those three features: large surface area, thin walls, and extensive blood supply. Now, you only need 2 of them for the marks here, but do try and memorize all 3 of those. That question is relatively common in coming up in the papers, so I'd make sure you know that. It would be very advantageous for you. Okay, part B, an investigation into the effect of water temperature on the breathing rate of fish was carried out. The results are shown in the table. So we've got the water temperature on the left going up from 4 to 26, and the average breathing rate on the right going up from 4 to 100. Now, you have to use the results to draw a conclusion for this investigation. Now, as soon as you are asked to draw a conclusion, you should be thinking, okay, the conclusion must relate back to the aim. So what is the aim? Well, it tells us in the stem of the question. It says the investigation is looking into the effect of water temperature, one variable, on the breathing rate of fish, the other variable. So the effect of water temperature on breathing rate. So therefore, our conclusion should mention water temperature and it should mention breathing rate. Okay. Now, the way to write these is you would write as the, in this case, we're going to put water temperature first because it appears first in the table. There's a more detailed reason why that is, but we'll keep it simple for now. So as the water temperature, now what's the water temperature doing in that table? It's increasing, isn't it? What is the breathing rate doing in that table? As the water rate temperature is increasing, it is also increasing. Okay. So as the water temperature increases, the breathing rate is also increasing. Okay. So we've got a nice relationship there, positive relationship, where as one goes up, the other one goes up. You could do vice versa. As the water temperature decreases, the breathing rate also decreases. That would be acceptable too. All right, so conclusions relate back to the aim. And in this case, we've got a direct measurement of the 2 parts of our aim, water temperature and breathing rate. That's actually what's in the table. Sometimes what's in the table is something different, and you still have to refer back to the aim rather than what's in the table for that one. But we'll come across examples of that another time. Question 12. Students investigated the distribution of some organisms on a rocky shore. Starting at the lowest tide level, quadrats were placed every 2 metres along a single transect line, and the number of barnacles and mussels were counted. The results are shown in the table. Okay, so we've got this low tide all the way to high tide. We've got 10 quadrats all the way along, and we've got the number of barnacles and mussels. Calculate the average number of mussels per quadrat. Okay, this is one of those questions. It's simple, but it's not easy. The simple thing we have to do is we have to add up all the numbers. Okay, so 60 plus 58 plus 55 plus 50. Okay, now many of you will be saying, oh, I know how to do this, Mr. White. It's not too difficult, but being careless is probably the thing you have to watch out for in this one here. Because if you are careless, you might miss a number or you might do something silly on your calculator, okay? So when we add up all those values together, we get, let me work this out in my head, 320. 320 is the total. However, to get the average, we have to divide by the number of values there are, and there are 10 values there. So 320 divided by 10 is going to give us 32. Okay, so that's how we work out the average there, 32 mussels. We add them all up and we divide by however many numbers we had there, which in this case was 10. Okay, describe how the reliability of the results could be improved. Now, we talked earlier on about VAVA, valid variables. Now we're going to talk about ReRe. So describe how the reliability of the results could be improved. RE stands for reliability equals repeat. Okay, we have to do some sort of repetition in order to improve the reliability. So you could simply say, repeat the investigation. That would be acceptable. The SQA would accept that. Okay, don't say repeat it. Say repeat the investigation or the experiment, and you'll get the mark. Or you could be more specific for this particular experiment and say carry out more transects, okay, or simply or use more quadrats, okay. Anything that suggests repetition of what we're already doing would help to improve the reliability of the results. Okay, so remember that reliability repeat. 12b, another group of students conducted a study of 10 rock pools along the transect. B part 1, to provide information about the levels of water pollution, samples of water were taken from these rock pools and examined for the presence or absence of certain species. Now, ding, 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 in my head, when I'm reading that and I'm seeing presence or absence of species, I'm immediately thinking of a definition. I know what name is given to these species. They are called indicator species. Indicator species, by their presence or absence, will indicate the level of pollution in a particular area. So, as soon as I saw presence or absence, I'm thinking indicator species. And if that's not ding ding in your head when you see that, then you need to learn the definition of an indicator species. Part 2, name the type of factors such as pH that can affect the distribution of organisms living in rock pools. There are 2 types of factors we have to consider. There are biotic factors, that's the living factors, and there's the abiotic factors, that's the non-living ones. Now, pH is not alive, therefore it's an abiotic factor. And again, if that's not immediately coming to you, you need to look through your list at the start of 3.2 of biotic and abiotic factors. You probably have a wee table or something similar in your notes that lists all of the biotic and abiotic factors. So you need to know those. And even if you don't know them off by heart, remember abiotic, non-living, biotic, living, and pH is obviously not alive. So therefore, it's going to be the abiotic factor. Okay, we're racing along towards the end of the paper now. We're on question 13, and we're asked here to look at an experiment that was carried out to investigate the effect of carbon dioxide concentration on the rate of photosynthesis. The rate of photosynthesis was measured by recording the mass of sugar produced per hour. Okay, we can see the carbon dioxide increasing along here, and we can see the mass of sugar increasing up this particular axis. Okay, what I'm interested in, when I see questions like this, I'm immediately thinking, okay, X is pointing to this flat bit of the graph, because that's going to be important later on. And I'm starting to sort of mentally think about that already. But A part 1 wants us to describe the relationship shown between the carbon dioxide concentration and the mass of the sugar produced. Now, this is a 2-mark question, you'll notice. So therefore, we're going to have to say 2 different relationships here. So let's start off with the first one, which is hopefully the more straightforward one. So when we're doing a describe the relationship question, where the question starts like that, you want to start your answer with as the, and then you just put the thing at the bottom here, which is carbon dioxide concentration. Okay, as the carbon dioxide concentration increases, and we can see the numbers are going up along the scale there, we then put a comma and we say the other thing. Okay, so that's the mass of sugar produced here. And what's happening to that? We can see the graphs going up. So that is also increasing. Okay, that also increases. That would get you 1 mark if you said that. However, we then say until, what's increasing until 0.08, okay? And sorry, that would get you the mark now. You have to say until 0.08 first, all right? And then what happens? If we look at X there, we can see that the mass of sugar produced levels off, okay? You have to say this part here to get the second mark, all right? So make sure you get both parts there. It goes up, then it levels off. You have to mention both in your answer to get the 2 marks there. Suggest one factor that could be limiting the rate of photosynthesis at point X in the graph. Well, when the letter is on the upward gradient of the graph, we know that the limiting factor is what is on this axis here. However, the letter here, X, is on the level part of the graph, okay, which means it's not the carbon dioxide concentration. It must be one of our other 2 factors, okay, which are that limit photosynthesis, which are temperature or light intensity, okay. Now, do we know which one it is? No, because we don't have any additional information. So we put down both there, okay? You actually would get the mark for just putting down one of the 2. The SQA would give you the mark for that, but I like to pop down 2, just a wee bit of flourish with your answer and show off what you know. If you're certain that you know it's both, well, you may as well put them both down, right? You spend 2 years studying this course or a year studying this course, and you may as well show off everything that you've learned. Okay, B part 1. Hydrogen is a product of the light reactions and is required for carbon fixation. Describe how hydrogen is produced. Now, hydrogen is produced when light splits the water into oxygen, which is our waste product, and diffuses out of the leaf, out of the cell, and hydrogen. And that hydrogen, as we know, is so vital for the second stage, so gets passed on to the carbon fixation stage from the light reaction stage. Okay, so you have to mention something along the lines of the water is split, or it splits the water, or something like that. B part 2, the sugar produced during the carbon fixation stage can be converted into other substances, such as starch. We have to name one other substance the sugar can be converted into and state its role in the cell. So again, hopefully we know the difference here between the two. So we've got our cellulose, okay, so sugar can be converted into starch and cellulose. It can also be used for respiration, but that's not relevant to this question. Its role within the cell is it provides structure within the plant cell wall, okay, the plant cell wall, okay, or you could say something like it makes up the cell wall, or it's a component of the cell wall, okay. Something similar to that would get you the mark there. Okay, moving along, question 14. Now, I like these questions when I see them. It gets me excited to see a 4-mark question because, you know what, it gives you the opportunity to show off all of that studying you've been doing. Okay, it's an open question about a particular area of the course, and you can write down everything you know. Okay, so in this particular one, we're asked to look at this food chain from a Scottish river and explain the role of the pondweed in the food chain and explain what happens to the energy at each level in the food chain. So let's kick off with the pondweed. The pondweed is at the start of the food chain, so therefore the pondweed is a producer. Okay, it's a green plant which photosynthesizes to produce its own food. Okay, and actually, if you didn't say producer but said all of that, you would get a mark there as well. The second part of the question says, explain what happens to the energy at each level in this food chain. Well, first of all, because the pondweed is a plant and it photosynthesizes, it needs to convert the light energy from the sun, and this happens in the chloroplast. It converts that light energy into chemical energy. Okay, and that chemical energy is in the form of it will be in the form of ATP, which we know gets passed on to the carbon fixation stage, but that's not relevant to this particular question. Now, that energy that we've made is then going to be passed on or transferred to the next level of the food chain. Okay, then we would say something like only a small quantity, let's say 10%, okay, or a small quantity. Apologies for my atrocious typing. A small quantity is passed on, okay, or used for growth would be acceptable as well there. You could say that 90% or the majority of energy is lost. You could say how it's lost. It's lost by heat, by movement, and by everyone's favourite, the undigested materials. Okay, so we lose a bunch of energy through those three. And then overall, as energy passes through the food chain, more and more is lost. So energy is lost at each and every stage of the food chain. Okay. Or sometimes we say the level, don't we? Rather than each stage, we say the level of the food chain. All right, so there's 7 different points. You could have put any one of those would have got you a mark, which means that you could have picked up the 4 marks for this question. As I say, you should feel excited when you see these questions because all that hard work that you've done, all that revision you've done, all that studying you've done, this is an opportunity for you to show it off in a big, extended, long question. So it should excite you rather than fill you with dread when you see these. If it does fill you with dread, that means that you need to be doing more work on memorizing your content so that these questions do have the opposite effect on you. And the other thing you can do, of course, is go through all the past papers and pick out the 4-mark questions or the 3-mark questions and work your way through them. Once you've done them all, they will hold much less fear. It's like anything in life, you face your fears and they become less terrifying when you do. Okay, so don't worry about those 4-mark questions. Question 15a. The increase in human population requires an increase in food production. Name a chemical in fertilizers that helps to increase the food yield. We've got a nice wee chemical inside our fertilizers called nitrates, which the plants, of course, use to build up amino acids into proteins. And then, of course, that plant protein is eaten by animals and passed on and so on. So we need plenty of nitrates within our soil for our plants to grow strong and healthy and give off a good food yield for the organisms that consume them. Part B, food crops are often affected by insects and disease. The table shows the average losses in yield caused by both in 4 crops in Scotland. So we've got wheat, barley, oats, oilseed rape, and insects and disease, and their average loss in yield. We have to fill in a bar graph here showing the label and the scale and then plotting the remaining bars. So I often see this as a mistake in a graph where people fail to put the correct label in this area on the graph. You're simply copying and pasting what is in the table. So average loss in yield and then in brackets percentage is exactly what you should be writing on the label there, okay? Don't try and shorten it, abbreviate it, change it in any way. Just write exactly what is in the heading in that table and you can't go wrong. In terms of the scale that goes up the side, now we look here and we see that wheat is on the second rung here, the second rung of that scale. So we go up to wheat, and this is insects, and we see that wheat for insects is 4. So we know that that's 4 here, which means this must be 2 and this is 0. And then each subsequent one is going up by 2, so that will become 6, 8, 10, 12, 14. And you'll do your scale all the way up to 16. And then if we look at our table, that works out perfectly because our highest value is 14. Okay. Then you have to fill in the bars. Now, I'm trusting, I'm not going to do this on the screen just now, but I'm trusting you'll be able to fill those in accurately. The key thing with the bars is to remember to nice sharp pencil, nice straight lined ruler, and making sure the tops of those bars are nice and straight, okay? Even just taking a few extra seconds to make sure in the exam would be good practice. In terms of the disease, we can see there's a couple of trickier ones. The 6, the 14, and 12 should be fairly straightforward because they're dead on the line, but the 7 is halfway between a line because there's no line there for 7. So that'll be halfway between our 6, which will be here, and our 8, which will be here. So we're wanting to do a line for the 7 value, which is for wheat. Okay, so that bar will be around about here. We're wanting to do that 7 value around about here. So right on the halfway point between the 6 and the 8. Okay, and be as accurate as you can when you're drawing these bar graphs, because you know what? You don't actually need any knowledge for this. It's just a skill. And if you practice it and you take your time in the exam, you'll be fine with that skill and hopefully you'll get the 2 marks. Okay. So just take your time, take a deep breath, copy the heading in the table as the label, make sure the scale matches up and can fit all of the values from your table into it, and then carefully plot your bars. Okay, question 5B, part 2. Identify the crop with the lowest combined percentage loss from these 2 causes. So which crop has the lowest combined loss? So we've got wheat with 4 and 7, that's 11. We've got barley with 7 and 6, that's 13. We've got oats with 8 and 14, that's going to be 22. And we've got the oilseed rape with 8 and 12, that's going to be 20. So the lowest combined there is going to be the wheat, okay? So our answer for this one is going to be wheat. Okay, so let's scroll down and get wheat in there. There we go. Okay, explain why it would be incorrect to conclude that the yield for each crop plant is affected more by disease than insects. Well, let's see why that would be incorrect. Affected more by disease than insects. So is it affected more by disease than insects for wheat? Yes, it is. Is it affected more by disease than insects for the barley? No, it's not. Okay, so because the barley is the exception to a rule, we cannot therefore conclude, therefore it's an incorrect conclusion, that the disease is affected more by disease than insects, okay? So barley shows a greater loss to insects than disease, okay? It is the exception. All the other ones show a greater loss to disease than insects, except the barley. Okay. Part 4, the total crop of the oilseed rape harvested was 140,000 tonnes. They calculate the yield of the oilseed rape that would have been produced if insects and disease had not affected these plants. So basically it's saying, what would it be if we hadn't had the loss? So let's see what the loss is. So it's 8 and 12, which is a total of 20% loss, okay? So we've got a 20% loss. Now, basically what we need to do is we need to work out what it would have been if we hadn't lost 20%. So the first thing we're going to do is we're going to get the 140,000. That's how much we do have. And we're going to work out now, this is if it's lost 20%, okay, if 20% is lost to insects and disease, that means what we have here is 80%. Okay, 140,000 is 80% of what it could have been. So the first thing we're going to do is we're going to divide 140,000 by 80. Now, why are we doing that? Because that will give us 1%. If 140,000 is 80%, then we divide it by 80 and we get 1%. So the 1% would be 1,750. Now, we're wanting to imagine there's been no loss. So if there's no loss, that would be 100%. So we're then going to times that 1,750 by 100, and we're going to get 175,000. Okay, and that would be amount that we would have if there was no loss there. Now, don't be tempted to do, I see this a lot in students, they try and add 20% to 140,000, okay? If you do that, you're going to come out with a slightly different value. I think it's like 168,000 tons, if I remember right. And that's obviously not the correct answer because you're adding 20% on top of the 80%. So that's 20% of the 80 rather than a 20% loss of the original 100%. So use the method that I've shown you here, work out 1% and then times it by 100. And that should keep you right and get you the right answer. Okay, over time, pesticides can build up in the cells of living organisms. What's the name of the term for this? This is just a simple definition, accumulation. Next one, if you don't know that one, by the way, look up your bioaccumulation in 2.5 and make sure you memorize that. Okay, sometimes a predator of the pest species is used as an alternative to pesticides. Name this method of reducing pest species. So the natural predator of an organism, the one that will kill it in the natural order of things, is called a biological control. Okay, because it's a natural predator, it's biological. And it controls the population of the pest species. So biological control. And again, that's a memory one. If you don't know what biological control is, look up your notes in 2.5 and memorize that. Do yourself a favor for the exam. Okay, question 16. We're almost there, folks. Hawaiian honey creepers are species of birds that have evolved from a single ancestor to have different shapes of beak. This is an example of speciation. For speciation to occur, a population must be split by an isolation barrier. Name one type of isolation barrier. Again, this is just memory work, right? So we're looking for geographical, ecological, or behavioural. Okay, you don't need to put, you don't have to put all of these, just whichever one you can remember. But it's useful to have one or two in the bag just in case your mind goes blank on the one that you have memorised. Okay, so yeah, one of those. The different mutations occurred in each subpopulation of honey creepers after a population was split. Name the molecule within a cell in which mutations occur. Mutations occur in our DNA. Name one environmental factor which increases the rate of mutation. Again, this is a straightforward knowledge question. It'll be radiation or chemicals for that one. Okay, again, if you're not sure about these two questions, go and have a wee look at the start of 3.6 and make sure you memorise those. Next question, suggest the selection pressure that could have led to the differences in the honey creepers shown in the diagram. So a suggest question, so we know that this is going to be a more challenging question. What is the selection pressure that led to the differences in their beaks? Now, what do they use their beaks for? Usually to get food, right? So why would they all have different beaks? Because initially they were probably all in competition for the same food, the same resource. And then over time, they evolved so that their beaks were different and they could access different foods, different types of food. And therefore, that would reduce the competition. So the selection pressure there is going to be competition, okay? And that competition was for food. And lastly, explain what is meant by the term species. Again, this is a straightforward definition one. You need to learn this one. Make sure you know it for your exam. The definition of a species is organisms that can interbreed to produce fertile offspring. And if they can't produce fertile offspring, they are not the same species. Okay, just fix my bad typing there again. There we go. Organisms that can interbreed to produce fertile offspring. This is a straightforward memorization one again, so go away and memorize that if you got it wrong on this occasion. Okay, I hope that was helpful going through the paper. Hope that you've picked up on a few things that you need to go away and work on and study before your exam, and best of luck when you come round to do that. See you next time.